Hello, everybody. Thanks so much for the opportunity to be here today. It's a real pleasure. I'm very sorry not to be able to be there in person, but it's a real privilege to be asked, and I'm glad to be able to contribute something, even if remotely. So thank you so much to the organisers for inviting me to speak today, and I wish you all the very best for an exciting programme over these couple of weeks. It looks like a really exciting group of people that are being pulled together to discuss a really important topic. So thank you, and I hope today's talk from me can help to contribute to that. So my name is Rory Gibb. I am currently an Excellence Fellow at University College London. I'm an ecologist, and I work mainly on the ecology and epidemiology of zoonotic and vector-borne diseases of various kinds, and their interactions with the environment and how kind of human, societal, and environmental changes are reshaping the dynamics and distributions and burden of many of these diseases that we care about. And ultimately, I'm really interested in how we leverage a better understanding of the ecology of pathogens to better support decisions that can help to reduce their impacts on health. So the goal of today's lecture is really to think a bit about how we define zoonotic disease risk, why that's important, and how that can help us to better think about how we could intervene. So as I said, my background is in ecology and in infectious disease ecology in particular, but over the years I've, I guess, become more interested in a kind of wider understanding of how zoonotic disease systems work and how ecosystems interact with people in order to shape the risks of infection and how environmental drivers kind of cascade through these systems. So I've worked a lot on rodent-borne diseases and various different vector-borne zoonoses, including things like dengue, Lassa fever, and CCHF, as well as kind of studying in more general terms some of the kind of macro scale patterns of infection. So I've been based at University College London for quite a few years. I'm also an investigator within the Verena Institute, so the Viral Emergence Research Initiative, which is a group of researchers that are kind of collectively thinking from various disciplinary perspectives about how we can better understand and predict viral emergence from wildlife populations. So that's a kind of sense of who I am and why I'm talking to you about this today. So the main objectives of the talk today will be to firstly describe the idea, the concept of hazard, exposure, and vulnerability, and how they contribute to risk as a whole, and think about how these different concepts apply to the transmission and emergence of zoonotic diseases. Kind of in the process of doing this, we'll examine some major contributions of disease ecology and epidemiology to understanding the risks of zoonoses. And then later on in the talk, we'll discuss a bit more about how socioecological interactions and feedbacks will shape pathogen transmission and spillover and disease outcomes. And this idea that risk is more complicated and shaped by some of these really complex interactions between social and ecological systems, and that we might need to understand those better in order to better intervene. So I wanted to start with some background context about why this is important. So the last decade has seen a really striking succession of health emergencies caused by novel or poorly understood infectious diseases, which originated in wildlife, starting with the Ebola epidemic in West Africa from 2013 to 2016, which had a really catastrophic impact on social and health systems within those kind of epidemic focal countries. Before that epidemic was fully over, we saw the emergence of Zika in Latin America, another WHO public health emergency of international concern, with severe birth defects associated with infection. And then we saw another major outbreak of Ebola, very complex outbreak, this time in the Democratic Republic of Congo. Then obviously the emergence of the COVID-19 pandemic and its extremely severe consequences, both in terms of morbidity and mortality, but also social costs as well as long-term economic consequences of this importantly ongoing pandemic. And during the middle of the COVID-19 pandemic, we then saw Mpox emerge, a rodent-borne infection, as a globally spreading disease. So what we're seeing in these contexts, right, is this sort of series of quite unexpected epidemics, which have proved to be incredibly complex to understand and tackle, and all of which involve non-human actors in the early part of their transmission cycle. So all of these have emerged from wildlife or have vectors, in the case of Zika mosquitoes, involved in their transmission. And simultaneously to this, we've also seen other disease crises occur. For example, the incidence of dengue globally has increased around 8-fold in the last 30 years and continues to rapidly escalate with a series of huge epidemics in recent years. So this has led to a general sense among the public and policymakers, and also in the scientific literature, that the frequency of epidemic events like this seems to be trending upwards. And so one of the questions here is why? Over the last decade or so, I think there's been an increasing understanding of the role of this sort of escalating anthropogenic impacts on the environment on many, many different dimensions of health. There was this sort of fantastic report by the Lancet Commission on Planetary Health, which was published in 2015, that in some ways established that sort of idea of planetary health, and included this great quote, which I really like. So this idea that we are deeply concerned that we have been mortgaging the health of future generations to realize economic and development gains in the present. And what they mean by that is, although we can see for these kind of globally aggregated indicators shown in the purple box, so kind of increasing population, we've seen declining poverty and increasing life expectancy on average worldwide. Those have come at the cost of a huge impact on the environment, a sort of massive extractive impact on many, many dimensions of the Earth system, which are starting to have long-term consequences, from climate change to impacts on ecological systems to water stresses, water shortages, and declines in biodiversity. And all of these have implications for health, including infectious disease. So the consequences of these sort of societal and Earth system shifts are to produce a whole series of proximate drivers that shape how pathogens spread among wildlife, between wildlife and people, and between people. So these include climatic drivers, including climate change and various different types of extreme events, ecosystem degradation and impacts on host and pathogen communities, urbanization and attendant inequalities in housing and services, as well as global mobility and connectivity through travel and trade, which influences how pathogens spread around the planet. And I won't go into this table in too much detail, but I really like this table from a paper published a couple of years ago in Nature Reviews Microbiology that breaks down some of the ways that these global change drivers can impact infectious disease risk at different stages of the disease emergence process, right? So from the leftmost column here shows how these different drivers in the table impact that initial process of emergence from wildlife or livestock into human populations. And then we see in the middle column how these drivers can impact the dynamics of disease within populations and consequent pathogen evolution. And then finally, in the rightmost column, how these drivers can impact the global spread of diseases. So, for example, when we look in the table, we can see climate change won't just impact one of these, but will impact them all simultaneously, right? So by changing how reservoir host species are moving around the planet and which other species they're coming into contact with, impacts on the transmission dynamics and susceptibility to infection, and then the geographical range of things like vectors that can, invertebrate vectors that can spread infection. So what I like about this table, and I would recommend digging into this table if you're interested in more detail, is just it kind of deconstructs nicely the many different ways that these drivers can impact risks. And this is something that we'll return to again and again through today's talk. So following the COVID-19 pandemic, discussions have really ignited across a whole range of different fields about how we could intervene to reduce the chances of something like this happening again. And there's been a sort of suite of reports published, commissioned and published following the pandemic that have reflected on lessons from the pandemic and what's next and what we could bring forward to the future, including reflecting on what worked and what didn't, both in terms of health response and also some of the discussions that ecologists have brought to the table around COVID origins, and also proposing the potential role of nature-based interventions, so kind of interventions at the level of ecosystems that might help to reduce the emergence of pathogens like COVID-19. One of the key things, that we see in these reports is that, authors who are coming from different sectors, whether that might be ecologists versus people who work more on the kind of clinical and health systems side, offer quite different perspectives on what the most appropriate ways to tackle, emerging epidemic and pandemic risks might be. So this is a nice figure from Rainer Plowright's recent paper on ecological interventions to reduce risks. And what I like about this is it kind of sort of breaks down quite nicely the sort of stepwise process by which pathogens emerge from wildlife into human populations to become epidemics. So the leftmost panel here shows the drivers of, we have pathogens that circulate within ecosystems and are driven by certain factors that then influences in turn how people might be exposed to those pathogens from wildlife. But then once pathogens have entered a human population, there are a whole series of barriers that determine whether or not that then becomes an epidemic and whether that epidemic then spreads widely enough to become a pandemic. And, from the perspective of COVID-19 and other emerging epidemics, if we view the history of emergence, all the way from emergence from wildlife all the way to global spread, we can kind of think about a whole suite of different interventions that could have stopped an infection like COVID-19 or perhaps Ebola from becoming an uncontrolled epidemic, or at the very least reduces impact after it had emerged. So just to give you a sense for some of those interventions that have been most widely discussed, one important one is predicting and preventing pathogen spillover from wildlife. So this is really thinking about, can we understand what's shaping pathogen diversity in wildlife and how it spreads, and pathogen prevalence and the likelihood that those infections could then pass on to humans. And then perhaps if we understand those drivers better, we might be able to use those to map kind of hotspots of risk and then prioritize resources, whether those are kind of health system resources or better surveillance or better kind of research in wildlife to kind of identify potentially risky pathogens before they emerge in a major way into human populations. But then more on the health system side, we can think about improving disease surveillance and rapid response, right? So ensuring that when outbreaks happen, they are quickly detected and those detections are quickly escalated to the right decision makers to ensure that responses are fast enough to contain emerging epidemics before they escalate to become the kinds of large-scale epidemics that have a severe health burden. And then on the social side, we're also, we can also think about how we can reduce the impacts of disease once it has emerged into human populations, right? So a lot of this is about reducing inequities in health and access to healthcare. For example, that might involve tackling the institutions that gave rise to such extreme vaccine inequity during the pandemic, or that continue to drive such extreme inequalities in access to high quality and affordable healthcare. So something I think is quite interesting looking at this is it highlights that how different people might prioritize these approaches, might rank their importance, depends in large part on how you view zoonotic risks and their drivers. If you work on the kind of health systems and vaccine side, for example, you might advocate strongly for the importance of those interventions. Conversely, if you're an ecologist working on the human-wildlife interface, you might focus a lot on those local socioecological factors that shape spillover transmission from wildlife, but might not place as much of an emphasis on some of those interventions that are really important at the later stages of emergence. So, I guess one of the kind of ideas in this talk really is, when we're thinking about how to prioritize interventions, we actually need to consider carefully what we mean by zoonotic risk, and how we are defining the risk of zoonosis or zoonotic epidemics. And this is actually kind of more of a non-trivial question than it might seem, because it in no small part depends on who you talk to and their disciplinary background and the sector that they work in. So for example, I come from an ecology and kind of disease ecology background, and often in disease ecology, zoonotic risk as an idea tends to be spoken about fairly synonymously with infection risk. And by infection risk, I mean the probability that you would be exposed and infected with a pathogen that is circulating in a wildlife population. And this figure is from Rainer Plowright's really fantastic concept paper from a few years ago. And this articulates really nicely the process of pathogen spillover, so that kind of infection process from wildlife to people, and the things that process depends on. So in the figure we can see at the top we have some distribution of reservoir hosts and infection intensity within those hosts. And then there are a series of processes intervening here, these kind of brown arrows, that determine whether those pathogens are excreted from the host, how humans come into contact with those, so the sort of exposure route. And then when you get to the bottom here, we have the susceptibility of the host to actual disease, given that they have been infected. But this sort of process of spillover, right, the infection of one individual, is in many cases really only the beginning. And so to give you a bit of an example of that, we can look back at examples of a few different outbreaks or epidemics to consider whether spillover risk really fully captures all the dimensions of zoonotic risk that we might want to capture. So here's the first specific example here is the Ebola epidemic in West Africa, which emerged in December 2013 in the forested area of Guinea, far outside the virus's original known range, which had previously been in Central Africa. And it will have spilled over most likely from a bat reservoir, since the most probable reservoir host for Ebola is still thought to be bats. But this was unexpected, right? This occurred outside its normal historical range. And one consequence of that was that Ebola virus spread undetected for several months before it was detected officially in March 2014. And then at that point, surveillance and treatment began to be rolled out. And then there was another delay then until August of that year, 2014, when the WHO declared a public health emergency of international concern. So there was a sort of slow response here, driven in large part by the unexpected nature of the epidemic, but exacerbated by a fairly stigmatizing international response, and histories of conflict and colonialism and weak health systems within the region. So all of these things contributed to the devastating impact of Ebola. It wasn't as simple as the spillover event. It was a much larger, much larger phenomenon than just that point of first infection. We can think about something similar for Zika virus. So Zika is an Aedes mosquito-borne flavivirus, which has a reservoir in primates and is spread around primates by mosquitoes. And it was actually first discovered in Uganda in 1954. And it's been known to cause human infections, but they were always thought to be fairly mild, and there was no sort of sense that it was a kind of major epidemic threat until it reached Brazil likely through kind of international transport in around 2013. And it actually spread, because it was so mild, it spread undetected for over a year within urban populations in Brazil, with relatively mild symptoms in a region that also experiences outbreaks of dengue and chikungunya. So it was not noticed for a while until 2015 to 2016, when there was a major epidemic, which was exacerbated by the El Nino conditions, as well as poor urban infrastructure and services and relatively weak health systems in some of these epidemic focal areas, which fueled a really rapid spread, which was aided by many of the same conditions that have fueled the explosive emergence of dengue in these regions. So climate change, social inequalities, issues with vector control. And this led to quite severe neurological complications in a proportion of cases. So around 3,500 infants were born with microcephaly, and in a certain proportion of adults, Guillain-Barré syndrome was observed. So this was a sort of much more severe epidemic than you would have considered if you had only looked at Zika in its pre-emergence phase, or the processes that cause Zika to spill over to humans in its kind of historical circulating regions. And the final example I wanted to give here is Lassa fever, which I've worked on a lot. So Lassa fever is a rodent-borne infection endemic to West Africa. It's a viral hemorrhagic fever. And in this map, we can see the kind of historical distribution of cases shown as points. So these are kind of locations where outbreaks have occurred, as well as the distribution of areas where we know that the virus does circulate in rodents, which are shown in grey. So Lassa virus was discovered in the 1960s, but genomic evidence indicates that it's a long-standing human pathogen in West Africa. And historically, Lassa had been characterized as mainly a biosecurity epidemic threat. But in truth, there's relatively little human-to-human transmission, and this is really an endemic disease of poverty. Populations in endemic areas are exposed at quite high rates, but many of those infections are asymptomatic. And we don't have a good understanding what it is that determines disease severity, but it is likely to include a whole series of biosocial factors, including things like age, pregnancy, childhood exposure, and nutrition. And the consequences of Lassa infection, as well as a relatively high risk of mortality for severe cases, can also include significant sequelae, such as hearing loss. So there are kind of disability consequences as well. So I guess what I wanted to get across through these examples really is that the sort of drivers of risk and the consequences of these infections stretch far beyond that initial spillover process. They're determined not only by the ecological factors that shape spillovers, but also the social factors that shape how epidemics play out, or that shape the consequences of infection, how severe disease might be. And so, if we're thinking about risk, we really need to sort of take this into account. So as well as the kind of direct morbidity and mortality we see for some of these infections, we see indirect mortality from disruption of health systems, a whole suite of different social consequences, as well as economic costs that often, with indirect costs that often vastly exceed the direct costs from ill health. So this pie chart here is an estimate of the economic impact of the Ebola epidemic in West Africa. And I think the thing that's really interesting here is that the kind of cost of Ebola-related deaths and direct economic costs, so the pink and blue there, make up around a third of the total cost. But a very large proportion of the cost was actually from impacts on the healthcare workforce, and also the cost of deaths from non-Ebola causes, which was caused by people not being able to access routine healthcare because of disruption to health systems. So the risks kind of extend much further than just that kind of point of spillover. Another aspect of risk that it's worth just considering is that zoonotic pathogens differ widely in their ecological and evolutionary characteristics. And the most important or influential drivers of risk to humans will also differ depending on the specific pathogen we're thinking about and its characteristics. So this is a nice framework for thinking about the stages of zoonotic emergence, so that the sort of different characteristics that zoonotic pathogens have. Stage 1, these are pathogens that circulate exclusively in animals. Stage 2, these are pathogens that can infect humans and cause primary human infections and disease, but don't normally transmit onwards from there in a human-to-human sense. So human infections are really driven by contact with wildlife. And then from stages 3 to 4, we're moving towards pathogens that are increasingly effective at spreading in a human-to-human transmission context. And so if we can think about some examples of where different pathogens sit on this, down at the bottom here for these stage 2 primary human infection pathogens, we have things like West Nile, Lyme disease, and hantaviruses. Stage 3, we have pathogens like Lassa fever, which can transmit between people, but don't do so very regularly. And then we get to stage 4, so these are zoonoses that can sustain long-term human-to-human transmission, things like SARS-CoV-2, Ebola, and dengue virus. And then at some point within this process, we have pathogens that become exclusive to humans, which, to be honest, I think sort of SARS-CoV-2 probably now fits in, at least in this kind of framework, as well as things like HIV and measles. But an interesting thing to think about from a risk perspective is that actually the majority of pathogens have the pyramid shape here reflects the idea that the majority of pathogens have more of this kind of stage 2 characteristic. Most zoonotic pathogens are not very effective at transmitting between humans. So it's really this ecological process that shapes spillover that is the defining process that shapes kind of disease emergence and incidence. And new pathogens like these are spilling over all the time. So this is a a global map of disease events, disease emergence events globally. So either newly identified infections, shown in blue, or diseases that have been identified in new areas, shown in red, since 2003. And this is doubtless non-comprehensive. So we are seeing, kind of spillovers from wildlife occurring all the time. But on the other hand, these sorts of epidemic pathogens like Ebola and Zika, or pandemic pathogens like COVID-19, these sorts of outbreaks are much rarer events. So far fewer pathogens have the kind of biological capacity to emerge and spread effectively like this, at least initially. And once they do emerge into human populations, the drivers of disease impact and burden, as well as spread, are predominantly social and economic, right? So we're shifting towards a different conception of risk that might prioritize, the sort of social and economic drivers over the ecological drivers. And this sort of rarity is reflected in the fact that sustained human-to-human epidemics and indeed pandemics are fairly rare events. So this is a map of pandemics since the start of the 20th century and their geographical origins. And what this shows is that there are around 10 pandemics since 1900, including several influenza pandemics, COVID-19, the plague pandemic, the third plague pandemic, which stretched forward from the mid 1800s, the cholera pandemics, and HIV/AIDS. But this sort of approximately 10 pandemics contrasts to however many millions of spillover events that are going on between wildlife and people all the time, right? So the factors that shape pandemic risk are a different set of processes than the factors that shape spillover risk. So what does this mean in terms of understanding the drivers of emerging disease risk and how best to intervene? So I suppose firstly, the drivers of zoonotic disease risk go far beyond point of first infection, and they encompass the many different factors that make individuals and populations vulnerable to disease. And it means that we might need to give a different weighting to these different sort of social and ecological contributors to risk, depending on the disease and the context. So if we're thinking about endemic zoonoses that predominantly spill over from wildlife, those ecological drivers are going to be incredibly important to think about from an intervention standpoint. If we're thinking about infections, once they have emerged, we start needing to think a lot more about the sort of social and political economic drivers of wider spread and disease impact and outcomes. So, the kinds of drivers that we might want to try and address may not be universal across all pathogens and contexts. So prioritizing how to intervene might require some flexibility. So in this next section, I'm going to talk about a sort of conceptual framework for how we pull all these things together into modeling risk and kind of decomposing these drivers. So this is a kind of classic risk modeling framework where we think of risk as being really occurring at the convergence of some hazard, which could potentially have an impact, the factors that cause people to be exposed to that hazard, so exposure, and then the factors that, given that people have been exposed to that hazard, actually make them vulnerable to its impacts. And the important kind of insight of a framework like this is that the factors that shape actual realized risks require the alignment of all three of these factors. One is not sufficient. You can't solely have the pathogen circulating within an ecosystem. That's not enough to produce a risk unless there are factors that make people exposed and vulnerable to that hazard. So they actually need to converge. So this is an example of how the IPCC have used this kind of framework to talk about climate change impacts on various health outcomes. So here we can think of hazards as being things like extreme heat or drought, floods, storms, and vector-borne disease. Exposure being kind of activities and livelihood factors that shape whether people are exposed to these hazards, like employment, housing quality, location, livelihoods, and then a whole suite of social factors that determine vulnerability to those hazards, including things like age, gender, mobility, healthcare, various pre-existing conditions or socioeconomic status. And all of those things determine realized risks. So a few years ago, we wrote a paper kind of applying and as one of the ways that we talked about zoonoses in this paper was to apply this kind of framework and describe just a simple way of sort of teasing these drivers apart for zoonotic risks, really highlighting this point that zoonotic risks emerge from the intersection between sort of hazards, so these are pathogens that are circulating within ecosystems, exposure, so factors that make people exposed to infection, and then vulnerabilities that determine the impact of those pathogens once they have actually infected people. And this is a neat conceptual device for thinking about how sort of social and ecological processes contribute to realized risks. And over the next kind of sort of half an hour or so, I guess we'll try and disentangle some of that and think about the evidence that we have for these different drivers. So on the hazard side, we can think about hazards as emerging from the diversity and prevalence of pathogens that circulate within local host communities. And so their geographic distributions in broad terms, as well as their sort of local abundance and behavior of hosts, spatial and seasonal population and infection dynamics of reservoir hosts and pathogens, as well as long-term population change in hosts, all of which can shape changing hazards. And we have exposure being activities that create opportunities for contact and transmission, things like land use practices, climate extremes, livelihoods, housing and infrastructure. And then individual and social determinants that shape the degree to which people and societies are vulnerable to infection. And that includes things like physiological immunity and susceptibility, as well as social factors such as health systems, access, inequalities, and things like pre-existing conditions and co-infection. One of the things I like about this framework conceptually is that it highlights some of the major challenges in our field, in that it kind of really clearly shows the disciplinary silos that exist within zoonotic disease emergence research. That really knowledge about how these risks form is fractured across the ecological sciences on one hand, that focus a lot on hazards and, to a lesser degree, factors that shape human exposure to those hazards. So these are sciences like disease ecology, community ecology, macroecology, and biogeography. And then on the other hand, really the sciences that give us insights into vulnerabilities and exposure in great depth are actually the social and health sciences. So things like epidemiology, clinical and veterinary medicine, and the social sciences. And this kind of this framework highlights that to kind of fully and holistically understand the risks posed by zoonoses, we require a kind of inherently interdisciplinary perspective here. So sort of putting One Health into practice. But the development of kind of large scale policy interventions and the search for kind of general patterns of risks has been really fragmented across these domains, and this has implications for how we understand how to act. But over the next sort of few slides, I'm going to talk separately about these sort of different sections, sort of hazard, exposure, and vulnerability, and our knowledge of them, and then think a bit later on about a kind of more integrated perspective on risk. So when we're talking about hazards, we're really thinking about research in disease ecology, right? Which has been sort of hugely productive over the last 20 years or so in really giving us a great insight into how infections transmit and spread within wildlife populations and the potential implications for human pathogen exposure. And this work kind of broadly spans scales of biological organization, all the way from macro scale biogeography and global patterns of infection, down to kind of viewing things from the level of individual pathogen species, as well as infection dynamics within ecological community contexts. And so over the next few slides, I'll talk a bit about some of these insights at different scales into what shapes zoonotic hazards. So firstly, at the biogeographic scale, we know that host and pathogen biogeography are deeply dependent upon one another. So these maps here show global patterns of parasite diversity in primates. The top set of maps here, the top map here shows observed parasite richness in primate species. So we can see there are clear kind of regions where there is a very high diversity of known parasites in primates, The middle row here shows an estimate of undiscovered parasite diversity, so those parasites that we think are probably there that we haven't yet discovered. And then the bottom row here shows an estimate of the sort of most likely zoonotic risk to people in those areas. But so the kind of key thing to take home from here, really, thinking about where primate diversity is highest, is that the diversity gradients of hosts and pathogens are closely correlated. And that's in part because of this idea that diversity begets more diversity. So if we have a higher diversity of hosts, this provides more opportunities for pathogens to co-evolve with their hosts. So a higher diversity of pathogens within high host diversity areas at this biogeographic scale. And in areas with a high host diversity and a high diversity of closely related hosts, and so this is really thinking about the tropics, where we've got a very high biodiversity and sort of high diversity of closely related species, there are also many more opportunities for pathogens to diversify through host switching, so through cross-species transmission from one species to the other. So this is a really nice paper by my colleague Greg Albury from a few years ago, showing that the likelihood that any pair of mammals will share viruses is determined by their phylogenetic similarity, so how closely related they are, which is this top left graph here, and also their geographical overlap. So effectively, species that are that have a sort of substantial geographic overlap and are more phylogenetically related are the most likely to share pathogens, and giving opportunities for those cross-species transmission events to happen, as well as the diversification of pathogens through that process. So it's this idea really that global biodiversity gradients give rise to global pathogen diversity gradients, and in turn, those give rise to global gradients in zoonotic pathogen diversity. So this paper by Barbara Han from a few years ago shows the global patterns of diversity in various, of zoonotic host diversity in various mammalian orders, sort of seeing that there are clear spatial patterns with a particularly high diversity of known hosts in bats in South America, for example, and a particularly high diversity of known zoonotic hosts of rodents in Europe and North America. So there's a certain kind of sense that zoonotic pathogen diversity is correlated to host diversity, and this might be one reason why we see, sort of higher rates of emergence of, spillover and emergence of many pathogens within tropical regions that are highly biodiverse. But on the other hand, just a caveat here that sampling biases are really a massive issue. And the more we look, the more it becomes difficult to disentangle the degree to which these sort of global patterns of parasite diversity that we know are shaped by true pathogens of true patterns of pathogen diversity, or are in fact predominantly artifacts of surveillance effort, so sort of discovery effort. This graph here is from a paper that I led a few years ago, where we looked at historical trends in viral discovery rates across various mammalian orders from the 1930s to the present day, and showed that actually those sort of shape of those trends really differs a lot in different mammalian orders, with a particularly couple of really notable observations that bats and rodents were both generally thought to be quite low diversity of viruses until sort of around the turn of the millennium. So I guess around the point of time when Nipah virus and then SARS-CoV-1 emerged, then we suddenly saw a huge escalation in virus discovery rate in bats and rodents, which have led the two of them to become the most virus diverse orders, or at least known to be the most virus diverse orders according to the data that we have. And the point here really is not that that's necessarily inaccurate, but the point is it's actually really difficult to know. So these sort of evolving sampling biases over time shape our knowledge of the mammalian virome, and we need much more systematic studies to understand better how pathogens and potential zoonotic pathogens are distributed in wildlife. And our understanding of risk might differ significantly depending on when we look or where we look. So one way of trying to fill those observation gaps is through predictive modeling. And this is some work that a lot of colleagues of mine have done extensively within the Verena Institute. And this is thinking about how we can leverage existing knowledge about host virus networks that we have observed and the ecological traits of hosts to predict those species that are most likely to be hosts. So this is effectively a machine learning problem that is one way of kind of predicting unobserved zoonotic hazard. And so what these sorts of studies will do is they will take a known network of host pathogen associations. So this is nodes will be hosts and pathogens with links denoting whether they've been detected in association with each other. Then we use the traits of those hosts, combine those with machine learning models, and then identify those traits that are associated with reservoir status and use those to predict those species that might be most likely to be hosts. And so this is a paper by my colleague Dan Becker trying to do this during the COVID pandemic to try and predict likely bat hosts of beta coronaviruses, and showed that in this top map here that there are predicted hotspots of unobserved coronavirus bat host diversity that we expect to be in Africa and Southeast Asia. But then they also use these models to try and predict the most likely bridge hosts of SARS-CoV-2. And you can sort of see in this table here a sort of list of candidate species that these models predicted to be most, like probably involved as bridge hosts in SARS-CoV-2's emergence process. So this is one way that predictive modeling can try and sort of fill some of those surveillance gaps and give us a better understanding of these large-scale patterns of zoonotic hazard. But we can also take a view of hazard at the level of individual pathogens and ask, what are the characteristics that predict whether a virus will infect humans? And could this help us to identify the kind of evolutionary potential pathogens that may pose an epidemic or pandemic risk? And this is an example here of a nice study by Nardos Malens, who's based in the University of Glasgow, showing that you can use genomic features extracted from the genomes of a huge diversity of sequenced viruses to predict the likelihood, quite accurately, the likelihood that those viruses can infect people. And what this figure shows is an example of using those models to then predict the probability of human infection across the left-hand panel. Here is a whole range of different coronavirus species, so species within the Coronaviridae family. And on the right here, across a whole range of SARS-related beta coronaviruses that have been detected in wildlife populations. And basically using these models to try and identify those viruses that are most likely to be able to infect people based on their genomic features. So trying to think about, could we identify those pathogens before they become a significant risk to people? But we also know that beyond this idea that you might be able to predict pre-emergence human pathogens, which is a challenging and contested field, we can use viral genome features in the same kind of way to improve our predictions of unobserved hosts in a similar way to what I showed you before for predicting bat hosts. So this is a paper that some colleagues from the Verena Consortium led on, showing that bringing in genomic features from orthopox viruses helps us to do a better job of predicting those mammal species that are most likely to host orthopox viruses. And so this tree shows mammal genera and the number of predicted links to different orthopox virus species. So showing that there are certain genuses that have a particularly high likelihood of hosting particular orthopox viruses, which many of which are important zoonoses. So we can think about kind of leveraging these kind of macro scale patterns of observed infections in wildlife to do a better job of predicting where those hazards might exist, but we haven't seen them yet, either in space or across phylogenies. Another kind of parallel and in some ways longer running set of work on this topic has been on the community ecology side, and really trying to understand at the level of host communities and their disease dynamics, how do anthropogenic pressures shape host species communities for multi-host pathogens and the dynamics of infection within those communities that might in turn drive changing risks for people. And so this is really a kind of disease ecology and disease community ecology perspective. So this is an example really of how land use change reshapes species diversity within ecosystems, including host and vector species that can have consequences for infection risk. So on the left-hand side, we're starting in a kind of least disturbed kind of primary vegetation habitat and gradually moving as we move rightward across the slide into progressively more degraded landscapes, secondary vegetation, agricultural landscapes, and then kind of cities. And what these boxes underneath with the circles show is the different colored circles represent different species. So it's effectively a kind of a cartoon of the species composition of these communities. And so on the left-hand side, we can see this kind of highly biodiverse community with lots of species, including quite a few rare species that exist at quite low density. But then as you move, kind of along this gradient and towards progressively more disturbed habitats, what we see is the loss of those more human sensitive and rarer species and their replacement within the community with fewer species, many of which tend to be kind of more common habitat generalist species that have a kind of faster pace of life and which are often more resilient to human disturbance and may live at higher abundances. So these are kind of quite profound community changes that occur during the process of landscape change, which can in turn have significant impacts on hazards for human disease. And the kind of major root of this, right, is through ecological community change. And so for around 20 years now, there's been a growing and important body of work on this idea of the dilution effect. So this idea that as biodiversity is lost locally from host communities, that less competent hosts, so species that are less capable at mounting a significant infection and then transmitting that onwards to other individuals, if those more competent hosts are more likely to persist as ecological communities change, then what we see is an increase in disease risk in these less biodiverse communities. So as land use becomes progressively degraded, the more competent hosts persist, then the less competent hosts tend to decline, and what we then see is an increase in infection prevalence within the remaining hosts. And so this slide here shows the findings from some of the kind of canonical early work in this field, which emerged from studies of Lyme disease in North America by Richard Ostfeld and Felicia Keesing, asking this question of why Lyme disease was surging in suburban areas. And it turned out through sort of elegant combinations of field and experimental work that this was a kind of community composition effect, that as you move into these more disturbed kind of forest landscapes, the density of very competent white-footed mice was increasing, and the density of some of these less competent hosts was declining, leading to increases in infection within the wildlife population and consequent increasing risks, hazards for infection for people. And this was proposed as a kind of more general mechanism within ecology, the dilution effect. So this proposes that as communities disassemble under disturbance, those species that persist are disproportionately likely to be more competent hosts. And so in turn, that pathogen transmission would be amplified in those areas. This was highly debated, probably one of the most debated in ideas in ecology over the last 20 years or so. But in recent years, I think a kind of more of a middle ground and a consensus has been reached around the conditions under which dilution effects occur. And they have basically concluded that dilution effects are a kind of a general ecological principle and a predictable feature of changing ecosystems, but they occur not in the context of natural biodiversity gradients, but in the context of when biodiversity is lost locally. So these figures are from a really nice meta-analysis by Fletcher Halliday from a few years ago, showing that when in these systems where biodiversity is lost, we see a dilution effect. So we tend to see increases in risk associated with those systems where biodiversity has been lost locally, so in a kind of disturbed context. And some of the work that I've done over the last few years has been thinking about, how we might use this to think about, a general understanding of zoonotic disease risks. So not only thinking about infection in general, which, the dilution effect really thinks about infection as a general ecological principle, but thinking more about how we apply this to zoonoses. And so this was a paper that I did a few years ago, where we looked at a really large database of ecological communities worldwide, and we were able to show that across many different species groups, that known zoonotic hosts, which are shown here in brown, tend to either persist or increase in human-managed landscapes. Their populations tend to persist or increase in human-managed landscapes compared to primary vegetation, which is represented here as a dotted line. Whereas non-host species within the same orders tend to decline. And we see these effects, these sort of filtering effects, most strongly for rodents, bats, and passerine birds. Whereas we don't, see such clear differences among carnivores or primates, for example. So, what this sort of suggests from the perspective of zoonoses is that there is actually quite a lot of heterogeneity in how we might expect these systems to respond, and that changes in disease hazards and risks to humans may depend a lot on the specific host responses to human pressures. And we also know that vector biodiversity responds in similar ways to local environmental change, right? So we see sort of changing availability of different types of breeding habitat for mosquitoes, which can lead to really significant changes in vector communities, which has implications for transmission. For example, many Anopheles species tend to be found in more rural areas, whereas Aedes vectors of major arboviruses are often affiliated to urban areas. And there was a really nice kind of demonstrative study showing a couple of years ago, using some disease surveillance in Brazil, that as you move along this kind of gradient of disturbance, so the human footprint on this graph here, from 0, which kind of reflects, very rural and undisturbed areas, up to 40, which represents highly urbanized areas, that, you see a shift in the assemblage of diseases. So these hazards are changing away from infections that are transmitted mainly by rural vectors. So these are kind of malaria and leishmaniasis, towards infections that are transmitted by urban vectors. So we see a change in hazards is reflected in this kind of shifting disease assemblage across different types of landscapes. So these are some of the ways that we can think, that ecology gives us insights into how transformations in landscapes can change infection hazards. But those are not necessarily sufficient for realized risk, right, in the absence of pathways for exposure and vulnerability. And so a lot of work on exposure pathways has been in kind of things like global change biology, anthropology, and human behavior sciences. So some of these kinds of drivers that we think about as impacting exposure a lot are things like land conversion and fragmentation, the role of livestock as kind of intermediate or bridge hosts, human livelihoods, or hunting and the wildlife trade as all being kind of factors that can influence exposure. And I'll talk briefly now about some of those pathways. So this is an example of a kind of, what one might think of as a kind of risky landscape, where we have a kind of mixture of kind of forest remnant in the middle here, and then some kind of agricultural system around the outside. And within the forest remnant, we might have some forest species that aren't found within the kind of human disturbed habitat. And conversely, we have the kind of human disturbed habitat with lots of livestock and synanthropic species. But then within these fragmented landscapes, what we have is a lot of edge effects, right? We have a lot of ecotones, these points where these ecosystems butt up against each other, and cross-species contact rates might be higher. And so this kind of underpins a lot of the theory for pathogen spillover and epidemic emergence in fragmenting landscapes. So this is a nice paper by Christina Faust from a few years ago, which basically built a kind of toy model to show, what happens if you kind of convert habitat from, a predominantly kind of green, forest kind of habitat shown in these bottom squares here on the far left, all the way to a predominantly human habitat. At what point do those edge effects peak? And what does that mean for the risk of pathogens spilling over? And what does that mean for the risk of pathogens, igniting into epidemics? And so what we see here in this red line is that edge effects peak right when you're kind of halfway through this conversion process of a landscape. And the kind of theoretical modeling that they did to kind of demonstrate what this might mean for emergence was to sort of show that up here on the top left, that the probability of spillovers occurring from the kind of forest population into the more kind of human population, what they call the matrix population, that probability of spillover peaks in that interim point where you've got the highest level of edge effect. But actually, the risk of large epidemics igniting peaks at much higher levels of conversion, because by that point you have a far higher density of humans or other kind of matrix hosts within the kind of within the brown habitat area. So epidemic sizes increase as you continue to convert, even though spillover risks might kind of decline at some point, as you effectively get rid of all of those species that would otherwise pose a hazard. So this sort of suggests then that, during these land conversion processes, there might be a kind of sweet spot, or whatever the opposite of a sweet spot is, a highest risk spot, where that combination of hazard and exposure kind of sort of matches just right, as well as human host population size, to create these large epidemics. So it's a toy. This is kind of a toy model, right? And there are a lot of complexities that would differ across different disease systems. But it nicely, I think, for me, it nicely highlights that there's a kind of complex interplay there of, kind of social and demographic factors on both the kind of reservoir host and the recipient host side that lead to outbreaks and epidemics. And these sorts of fragmentation processes are really relevant to thinking about many spillover and epidemic pathogens, including things like Ebola, zoonotic malarias, many mosquito-borne arboviruses, and henipaviruses. As well as this point that, land use change worldwide proceeds through habitat fragmentation. This map here shows changes in patterns of fragmentation over the last sort of 20 years or so, and showing how, in many areas where we're concerned about zoonotic risks, fragmentation has been increasing. So, these landscapes are expanding in the Anthropocene, and that may have implications for exposure to these. Another important conduit for exposure may be the role of livestock, especially within these kinds of fragmented landscapes. And a good example here would be something like henipaviruses. So this is hendrovirus and Nipah virus, which are bat-borne infections that cause severe encephalitis with a high case fatality ratio. Both can spill over directly to humans, but also involve domestic species as major bridge or amplifying hosts, pigs in the case of Nipah virus, and horses in the case of hendrovirus, that effectively act as a conduit for infection from bats to people. And we can think about the many different epidemiological studies into livelihoods that give us an insight into those sorts of livelihood processes that influence exposure to pathogens. And there's extensive evidence across many, many different systems for how livelihoods such as agricultural livelihoods or kind of forest-based livelihoods or hunting might impact exposure to infection. This is just one nice kind of meta-analysis that I've picked out as an example, published a few years ago, showing that across a whole range of different land use types here on the left and different diseases on the right in Southeast Asia, that if you are an agricultural worker, so someone who is exposed to agricultural systems regularly, you have a much, much higher odds of being infected with a whole range of different pathogens than you would do if you are a non-agricultural worker. So exposure to agricultural livelihoods really does predict exposure to a lot of these pathogens will be circulating in these systems. But the key point here is that you are more likely to be exposed if you are in that group rather than the non-agricultural worker group. So it is not simply the hazard that produces the risk, it is the interplay of the hazard and the exposure. And the final and really important part of this puzzle is vulnerability, right? And so kind of for time considerations, I won't go into as much depth on this as I want to focus, I wanted to focus in this talk really on how ecology kind of plays into this picture. So I've spent quite a lot of time talking about hazards. But I think it's really important to stress that vulnerability is in many ways the key determinant of the impact of these infections once they actually reach people. And sort of social inequities, as well as kind of various biosocial and physical factors determine the impact of zoonotic pathogens. And ultimately, without many of these, we wouldn't be seeing the impacts that we do. So these include things like immunity and pre-existing conditions, susceptibility of varying kinds, social determinants of disease outcomes, stress, as well as access to health systems. And as I say, I won't spend as much time talking about this, suffice it to say that epidemiology and social sciences give us great insights into many of these different drivers and why they're important. But I do want to focus on kind of one in particular because it came up so much during the pandemic, right? Which is health systems access to high quality healthcare and the right kinds of healthcare products to reduce risks. So access to vaccinations and point of care diagnostics for emerging diseases are both really, really important, right? So point of care diagnostics can help to ensure prompt and appropriate treatment for infections that are more likely to respond positively to treatment if they're caught early, right? So for something like Lassa fever, the earlier you treat it, the more likely you are to prevent mortality or severe sequelae. And so, it's really, really important to ensure that the communities that are most at risk have that vulnerability reduced as much as possible by ensuring that they have access to kind of rapid diagnostics. And as a further kind of more basic aspect of this, access to high quality healthcare services that are affordable, as well as vaccines. And so, vaccine inequity obviously continues to be a severe problem. Around 30% of the world's population haven't yet had a single dose of the COVID-19 vaccination. And this nice study from last year shows they estimated that in the 20 low and middle income countries that they studied, that more than 50% of the deaths that occurred may have been averted with either higher or earlier availability of vaccine doses, which is a really damning indictment of the system of vaccine distribution that we saw during the pandemic. So from here, I wanted to spend a few minutes, as we kind of move towards the end of this talk, reflecting a bit on the benefits and limitations of a framework like this and what it highlights to us in terms of what we know and don't know about zoonoses and how we might intervene, as well as sort of future pathways, kind of where we need to go next. So for me, this approach to thinking about zoonotic risk is helpful in no small part as a kind of device for clearly thinking about where the different processes, social and ecological processes and their interactions contribute to realized risks, and to thinking a bit about where those knowledges exist within the academy, within the kind of scientific knowledge base, and across disciplines. And I think so this for me at least helps me to very clearly kind of think about not only the knowledge that exists on these different drivers of risk, but also where interventions that are targeting different levels of the system might actually be acting on risk, and how we might think in a more joined up way about interventions that kind of prioritize these in a kind of joint way rather than just tackling one at a time. It also shows, I think, that we know, hopefully the very brief overview I've given you for the last half hour or so shows that, we know a lot about zoonotic risks already, right? But what we also know is that knowledge is really fragmented across the ecological sciences on one side and the kind of health and social sciences on the other. And this offers a kind of challenge to operationalizing this view in a more integrated way to design, for example, interventions that might target multiple levels of systems at once. And we really need to better understand those interactions better. Some of the other kind of challenges of this framework that I've reflected on a lot over the last few years are its kind of lack of dynamism. I think it has a certain kind of static property to it that is not necessarily useful for thinking about how the sort of dynamic processes by which diseases emerge. And it also, to some degree, inherently frames these three drivers as independent from each other, as opposed to integrated. And I guess I'll kind of talk a little bit more about that in a few minutes. And it also highlights that these disease systems are very complex and they're often poorly observed, and actually disentangling these drivers, hazard, exposure, and vulnerability, can be really tricky. So I'll give it a couple of examples for these limitations, and then we'll think a bit more about where to go from here. So first is this point about dynamics of change. I want to kind of give an example of of the emergence process and how we might think about hazard and exposure and vulnerability in this context. So this is an example for dengue, or it could be equally applied to other epidemic emerging arboviruses like chikungunya or Zika, or some of these kind of early stage emergence viruses like Mayara virus. So along this kind of stage of emergence, we have on the far left here sylvatic cycles of dengue virus transmission that's still going on within primates, and that occur in a kind of primate to mosquito to primate cycle within forests, in rainforests in Africa and Southeast Asia. And then we have what they called in this kind of nice paper from 2011, the kind of zone of emergence, these areas, again, thinking about fragmentation, where these sorts of forest landscapes are intermingled with human settlements, often in rural areas, and those people might become exposed to infection through these kind of vectors that act as bridges, so that will bite primates and then will bite people. And then ultimately, what we saw with the emergence of dengue and chikungunya and Zika is that, and yellow fever much earlier in history, is that if you have infected people moving regularly enough from those zones of emergence towards bigger built-up urban centres, you can see urban transmission cycles establishing with effective transmission among humans via mosquitoes, especially Aedes aegypti and albopictus, leading to these runaway epidemics and ultimately, especially in the case of dengue, to this kind of worldwide, extremely rapid spread that we've seen over recent decades. And so I guess it's interesting to think, I've thought a lot about this in terms of sort of, how we might apply a sort of hazard and exposure and vulnerability framework for thinking about, like an emergence process like this. I think the main thing that I want to highlight here is that the further left we are in this picture, right, that the more, our risk is really, really shaped by the hazard, right? When we're talking about the emergence from sylvatic cycles, we're really thinking about how viruses are distributed within primates and mosquitoes across forests, and thinking about exposure, those points at which, people within these rural areas are coming into contact with those circulating viruses in wildlife populations. But as we move further to the right, as we progress further within this emergence process, what we're seeing is the increasing importance of social vulnerability in determining how disease is distributed across the human population when you move further into this kind of established human-to-human transmission cycle. So we really sort of, thinking increasingly then about, vulnerability and social determinants is becoming increasingly important as we move into diseases becoming a progressively sort of human pathogen rather than a spillover pathogen. And it's important to kind of take that and, to sort of take that insight in mind when we're thinking about how we might intervene at different stages of disease emergence. Another challenge for this sort of framework from a more operational perspective is that the disease outbreaks that we observe are a product of hazard and exposure and vulnerability converging to produce realized risk, which I've circled here in red. And so we observe these, right, and we record them in surveillance data. But that observation process is partial and biased. We don't see all of the infections and disease cases that occur, particularly in low and middle income settings where access to healthcare is very variable. Often we only see a subset of them that are particularly severe, or in people that have better ability to access the kinds of clinics where these infections might be suspected and be able to be diagnosed. And ultimately then some proportion of those may get reported to case surveillance systems and then make their way into our disease incidence or outbreak or kind of outcome data that we have to study. And so this map here shows the sort of distribution of CCHF outbreaks across Africa over the last 70 years or so, just sort of showing that it's very sparse and patchy, and we know that a lot of that is driven by spatial bias in reporting. And kind of what that means is that the data that we have are very patchy. They are already the outcome of a complex underlying process that we don't understand very well. And then that is further kind of confounded by the observation process, which means that it's really hard to kind of reverse engineer from those data and go back and go, okay, what was driving these outbreaks? Was it, predominantly vulnerability? Was it predominantly hazard? And so it's very difficult for us to kind of weight the contributions of those different drivers to ultimate patterns of disease. And this was something that was highlighted in a project that I recently led, a large consortium data synthesis project, where we were trying to explore the detectable fingerprint of anthropogenic drivers on the geography of outbreaks for 32 emerging diseases worldwide. So we can see close to 60,000 unique outbreak events across the globe, shown here on this map. And applying a kind of standardized analysis framework to this whole set of data across all the diseases, what we found predominantly across all diseases was that the most consistent driver of the known geography of these emerging disease outbreaks is proximity to health centers. So the closer you are to a clinic, shown here in these graphs here at the bottom, the more likely you are to detect cases, and that effect can be quite extreme. Sort of an average of around a 30% decline in your odds of outbreak reporting for each additional hour's travel time to health facility. So you're talking about quite a steep surveillance bias, and these really structure our understanding, make it very difficult for us to go back and ask what those principal drivers are. And this is compounded by system complexity, right? So within this study, across all diseases, what these networks show is the drivers that we could detect. The size of the node reflects the number of diseases that driver was detected for, and the connections between the nodes and their weights reflect how many diseases shared those drivers in common, either for all diseases on the left, for directly transmitted zoonoses, or vector-borne zoonoses further to the right. And what we found was that these drivers were detectable, but the main detectable drivers seem to be detection biases, so access to health centers and being in a city. But then we could also detect very strong effects of heterogeneous vegetation and forest cover, as well as some effects of climate change. But these are kind of compound drivers, right? And these are complex systems. It's actually very difficult to know whether, for example, fragmentation is acting predominantly by reshaping ecological communities and so driving hazards, or whether it's predominantly acting on exposure and sort of changing the likelihood that people will be exposed to pathogens. So we really need to find other ways rather than these kind of large scale data sets that we'd have to tend to work with to better understand and disentangle these myriad contributions to risk within zoonotic disease systems. So this sort of really highlights, right, that the effects of these contributors to risk are not independent or linear, but they kind of shape each other and they interact, and they're all determined by historical forces that act on all of them. And so having this kind of slightly disarticulated framework for thinking about risk, which is replicated within disciplinary silos, means that we don't have as joined up a picture as we need to. And this is a paper that I've led that's kind of currently in review, where we're just sort of trying to take a bit more of an expanded view on this framework, and think about how socioecological interactions and feedbacks actually create kind of linkages between hazard, exposure, and vulnerability, and how these are all determined simultaneously by these larger scale drivers. So just to kind of give you a general sense here of what this is showing. So we have hazard, exposure, and vulnerability and their convergence driving risk. But there are many ways that hazards can shape vulnerabilities. So hazards, a high diversity and prevalence of hazards within the environment might in fact impact individual susceptibility to disease through co-infection or past infection histories. It might also impact population vulnerability through having kind of significant disease impacts on economies and health systems. And then, in turn, those population impacts can further shape individual susceptibility to disease. But increasing economic vulnerability can also drive exposure through kind of creating economic imperatives for land use change or wildlife use and hunting, as well as impacting things like housing or water and sanitation infrastructure that make exposure more likely. And in turn, those exposure activities can, for example, things like land conversion for growing crops, can create new niches for biodiversity and reshape hazards, and so kind of creating feedbacks within these systems, right? So hazard drives vulnerability, vulnerability can drive exposure, exposure can drive hazard. And increasing levels of exposure, for example, through wildlife hunting, might in turn reduce vulnerability, for example, through improving people's nutritional status or their household income. So this kind of highlights really that we do need to start building better links between the different subdisciplines that work on this problem. This kind of expanded view highlights that we need to think about putting these pieces of the puzzle together if we want to understand how kind of patterns of human risk and burden emerge from the intersection of social and ecological systems. And this is especially important when we're thinking about interventions, right? Where, if we're thinking about an intervention like, land use change, sort of land use regulation, we need to think in quite a holistic sense about the many different ways that that intervention might propagate through the system. For example, if tightly regulating land use change might increase social vulnerabilities by reducing people's income, even as it reduces hazard by mitigating biodiversity change. So these impacts might be compound, and we need to start thinking about those interlinkages in a deeper way. So for the last kind of section of this talk, I really just want to talk about some of those feedbacks and what we might do about it. So here's just a sort of few examples. Here we can see how vulnerability and socioecological feedbacks can kind of shape disease risk. This is a nice study of data from the Brazilian Amazon showing that increased deforestation drives increasing malaria through producing the right kinds of habitat for vectors to survive in and bite people. But actually that when you see an increase in malaria burden, you see a decline in deforestation, because that increasing burden of malaria reduces people's economic activity and inclinations to settle in those areas with a high malaria burden. So there's a kind of negative feedback loop here, that this causal modeling approach that they took in this study allowed them to disentangle clearly within the Brazilian Amazon. And we can see similar sort of interactions in how socioeconomic disparities and vulnerabilities can shape the hazards that emerge from sort of climate forcing on disease systems. So this is some work that I did on dengue that was published last year on the role of hydrometeorological extremes, so drought and rainfall excesses on dengue. And what we were able to show was that within Vietnam, with some work with some fantastic colleagues in Vietnam, we were able to show that long-term droughts were generally predictive of increasing dengue risks, as we sort of see here on the right, with sort of increasing risk under real severe drought conditions. And we think that's likely to do with people storing water in their homes in response to drought, which produces breeding sites for mosquitoes. But what we found that was that effect was particularly strong for certain households and not others. And what we found was that households that had a high access to improved water supply, so this is from the piped water network or from drilled wells, actually had a reduced risk of dengue during drought periods compared to households that had a low to intermediate access to improved water. And this is likely because they weren't needing to store as much water at home. But under conditions of severe drought, that relationship flips, and we see a decline in risk in those areas where people have very low access to improved water, and a very sharp increase in risk in those areas where people have high access to improved water, potentially because those people who are able to, who do have high access to improved water, would still be able to store water under those conditions, whereas those with low access to the piped water network would switch to bottled water. So this sort of highlights, right, that socioeconomic disparities have quite complex effects on the climate-driven hazards for infection. And another factor we can think about is how comorbidities and co-infections influence disease outcomes. So whether an infection leads to significant morbidity or mortality is shaped by various biosocial factors, which include comorbidities and co-infections. One of the best examples of this is how the COVID-19 pandemic had a particularly strong impact on marginalized communities in many countries that had higher rates of pre-existing conditions, as well as a higher likelihood of being exposed to infection. But the kind of circulation of multiple pathogens through, sort of multi-pathogen hazards can worsen disease impacts overall through not only kind of the interaction of pathogens with each other, such as arboviruses, but also by causing delays in diagnostics. So in areas where there's a high circulation of malaria and Lassa fever together, many Lassa cases are misdiagnosed as malaria and treated as malaria for a while until it becomes clear that they're not responding to malaria treatment, by which point Lassa becomes the diagnosis of last resort. And often in that case, it's fairly late for the available treatments to be as effective. So this increases the likelihood of mortality. So we see kind of complex impacts of the co-circulation of multi-pathogen hazards on vulnerability. And one final example would be that activities that drive exposure, such as hunting of wildlife, are often really important for food security in many communities. So this is a study by my colleague Sagan Fryan in Nigeria, in Cross River, showing that consuming rodents is really important for food security, even though in this area rodent consumption can also be associated with a higher risk of being exposed to various zoonotic infections. So I guess reflecting on all of this, what are the kind of next steps, and kind of what can we do to bring all of this knowledge forwards into thinking about how we might intervene better? One of the things we've learned, from trying to kind of tackle these questions in a multi-disease way is that interventions will often need to be tailored to the specific ecologies of particular diseases or regions, working with the local stakeholders and affected communities who really understand what those risks and challenges are. And that global kind of one-size-fits-all solutions for zoonotic risks might be hard to come by. Secondly, the drivers of risk are never independent from each other, but they're shaped by feedbacks, and we need to study those linkages much better to understand how different interventions or environmental changes might propagate in complex ways through systems. Ecological interventions are likely to be useful for many pathogens, especially those that spill over regularly from wildlife. But they might often need to be system specific, and they are likely to be complex to implement and evaluate, and so might operate on a much slower timescale than social interventions to reduce exposure and vulnerability. And so combining these approaches, rather than thinking about them independently, combined interventions that might include some ecological interventions, but also social, many different social and health system side interventions, may be the most effective in the long term. And lastly, the kind of the drivers of biodiversity loss and the drivers of disease risk are they share many kind of upstream forces that produce them. So here we're thinking about global capitalism, patterns of inequality worldwide, extractive industries, and power disparities, as well as climate change. We need to think in a systemic way about what those drivers are and how they simultaneously impact hazard and exposure and vulnerability in many affected communities. And I guess this just sort of highlights that we do need kind of truly transdisciplinary thinking in order to to sort of design interventions that could work effectively. So obviously the One Health paradigm really stresses this idea, right, that we sort of seek to see ecological health, animal health, and human health as interconnected, and that interventions to prioritize one of them should prioritize all of them, right? And this is reflected increasingly at the sort of highest levels of health policy and in things like the One Health quadripartite. But this highlights that in order to actually operationalize this knowledge, we really need much more of an interdisciplinary team science approach to do this. So this example here is the kind of parable of the blind men and the elephant, right? So we have four blind men who are each trying to say what this elephant is, and they each touch it in a different place, and they come to a different conclusion about what it is because they can't see the whole. So the person that touches the nose says, oh, I think this is a hose. The person that touches the leg says, I think this is a tree. The person that touches the body says, I think it's a wall. And the person that touches the tail says, I think this is a rope, right? And we can think about this in terms of the various different disciplinary perspectives on zoonotic risk and how to tackle it, across epidemiologists, clinical and veterinary medicine, social scientists, and ecologists. We each take different views. And we see sort of similar things right across different sectors, from health, agriculture and food systems, environment and climate, or business and pharmaceutical. And bringing those things together in a kind of team science-based way perhaps gives us an opportunity to understand them better. So I just wanted to give kind of one little example of a study that some really fantastic work that I'm a kind of consultant on, which is a study that's being led by a brilliant team at the Uganda Virus Research Institute in Entebbe called the CORI Uganda Project. And this is a study that's really focused on combining ecology, epidemiology, and the social sciences to identify those key local drivers of risk and how best to intervene. And And in practice, this has involved simultaneous deep sampling of humans, livestock, vectors, and mammal communities around 6 conservation areas in Uganda, doing sort of really, really deep epidemiological and social science work alongside that, and pulling together a data set that we hope, with some analytical care, will enable us to kind of better disentangle those contributions of circulating pathogen hazards in wildlife from the factors that shape exposure, like land use change, from the kind of entrenched social factors that might influence vulnerability to infection. And so, globally, on the kind of global side, there's been a lot of advocacy recently for kind of post-pandemic globally regulating to reduce zoonotic emergence at source, this kind of idea of primary prevention, and more tightly regulating kind of some of these hypothesized general drivers of emergence, like wildlife trade and deforestation. I think currently there's little evidence that this would be consistently effective across all diseases, and it doesn't take into account this idea of interactions and feedbacks among drivers that much. But there are some really important insights in here, right? Both about health system strengthening and the importance of poverty alleviation, but also the importance of improving kind of One Health surveillance of human and wildlife populations in those at-risk areas. And finally, just sort of making this point that none of these interventions need to be exclusive, and there is a lot of work that can be done to reduce vulnerabilities to spillover and epidemics on the health systems and health systems governance side, both internationally and within countries, from stronger leadership and coordination, surveillance and alert systems, and improving access to finance for affected countries and communities, as well as preemptive investment in vaccines and therapeutics, and importantly, enshrining that those become more equitably distributed in the event of future epidemics. So, to conclude, disentangling this idea of zoonotic risk into hazard, exposure, and vulnerability, I think, has the benefit of giving us a clearer picture of what we actually know already, and how human activities might exacerbate or mitigate risks through those various different pathways. But it also highlights that although we know a huge amount about zoonotic risks and how to address them, that knowledge is really siloed, and that this kind of idea of joined up research to kind of tackle those interdependencies better is relatively new. And that there's a real need for kind of these interdisciplinary team science-based approaches with folks from a whole range of different disciplinary perspectives, bringing that knowledge together within shared studies to kind of help disentangle these problems in more depth. And finally, to conclude, I think we really need to consider not ecological and social drivers as independent phenomena, but really think a great deal more about how they interact to shape infectious disease systems, because that will help us understand more about the most effective weak points in those systems where we could intervene to reduce realized risks. And with that, I'll say thank you so much for listening. It's, as I said at the start, it's been a real privilege to be invited, and I hope that I've been able to contribute something useful for you all, and I wish you all the very best with this program. So I'm very happy if anyone has any questions that they would like to reach out to me to find out more, or to talk about specific things that you heard in this. Feel free to drop me an email, or you can message me on Twitter. And just to drop a few acknowledgements, which are inevitably incomplete, because my thinking on this has been determined by interactions with so many fantastic people over the years. But particular kind of thanks to my colleagues at UCL CEBA and the people in Nature Lab, the Viral Emergence Research Initiative, and a suite of individual people who've been very influential and supportive of me over the last few years. So yeah, thank you all for listening.